Am I audible?
Yes, yes, very much audible.
Okay. So the next presentation we have is of our Chief Organizing Secretary, Dr. Siddhartha, currently joined as teaching assistantship. Hello, am I audible?
Yes, yes, audible, very much audible.
Okay. So the next presentation is of our Chief Organizing Secretary, Engineer Siddhartha, currently joined teaching assistantship cum PhD scholar in Dr. B.R. Ambedkar National Institute of Technology, Jalandhar, Punjab. He has recently worked on tissue engineering and biomaterial sciences. He received undergraduate degree in biotechnology from MMDU Maulana and post-graduation degree also in biotech from IK Gujarat Punjab Technical University, Jalandhar. He was also worked on acute dermal toxicity and decontamination study of carbon composite in Sprague Dawley rat models since 2021. He has more than 3 years of research experience in vivo study in nanobiotechnology, radiation, excessive wound healing, dermatotoxicity study, and radiation decontamination study. He was working as research intern in Institute of Nuclear Medicines and Allied Science, DRDO, Delhi, National Cancer Institute, Kolkata, National Institute of Biologicals, Noida, as well as ICTC Lab, MGM Medical College, Jamshedpur. He received PhD fellowship given by MHRD 2022, Best Young Scientist Award in year 2021, Shaurya Bharat Award in 2021. Engineer Siddhartha has also honored by various science societies as MIAZ in earlier years and currently life member of IAZ, Vijnana Bharati, International Association of Engineers, and All India Council of Technical Skill Development. He was reviewer of biointerference research in applied chemistry and reviewer management system, board member of International Journal of Creative Research Thought Grants. He has excellence in work in research activities and published various national and international research papers, review articles, and book chapters, including 30 plus review articles in reputed journals, which are available online too. Now, over to you, sir. All the best.
Thank you so much, Bhavna ma'am. I hope my screen is visible.
Yes, it's visible, sir.
Thank you so much. Now I would like to explain one very hot and very different area in biotechnology, that is bone tissue engineering and radiation prevention and decontamination in space engineering, as in astronauts. What is the major work in astronauts or space in bone tissue engineering or biotechnology. I am just talking about what is biotechnology for students of UG and PG, which is just joining recently. Biotechnology have actually many areas like food and nutrition, marine and aquaculture, industry, agriculture, environment, technology, medical, health, and diagnostics. It's also in biotechnology, they have two type of researches are available. That is primary research and secondary research. Primary research are three types. First one is the basic research like cell culture or normal lab works like biochemistry, genetics, or animal study. And next one is clinical research, that is phase 1 trial, phase 2 trial, phase 3 trial, and phase 4 trial. Mainly four types of clinical trials are there. And third one is epidemiological research, that is experimental or observational research. And second type is secondary research, that is only for meta-analysis or make a review paper. Now, my biotechnology broad area is medical and health diagnosis, and in that field, I am worked only in tissue engineering and biomaterials. That is from only waste product materials, and I want to this type of things are worked linked with space biomaterials, like these are also three types: habitation, astrobiology, and biomedical. In the field of habitation, that is only checked habit, suits, and coating. This type of worked in ISRO, India. But the other two types of work, astrobio and biomedical, this is like contamination types work and wound dressing, pharmacological work. These types of work are by only NASA or European Space Station or Russia Space Station. Only these three are worked on biomedical, like wound healing. And I only worked in tissue engineering. The main area is osteology, that is make a scaffold and the prevention of decontamination from radiation, that is only gamma ray. And I am worked done already in this type of work was done in DRDO in Mars. First thing ki why introduction, radiation exposure and contamination scenario. Why we can work on radiation exposure? Actually, we can see our conference theme is physiochemical, biological, radiological, and nuclear agents in human health. So first one is accident nuclear event. Like you can see in the last 50 years and hundreds years ago, that is Nagasaki and Hiroshima, we can see the nuclear events. And we can also see the one more in Delhi, Mayapuri, cobalt exposure. And next one is occupational exposure, that is like in normal industries waste. That's why this is the very important scenario for contamination by biomaterial waste product. Some types of biomaterials we can discuss, that is based on only carbon composite, that is graphene oxide, reduced graphene oxide with silver nanoparticle, reduced graphene oxide with silver nanoparticle, and reduced graphene oxide with zinc oxide, and many more. These biomaterials also worked as antimicrobial property and as making a scaffold, that is in teeth implants, biomarkers and oral cancers, hydrophobic property. These are also work in a bone regenerations and increase the activity of antimicrobial property. First thing we can check ki all biomaterials mainly worked in for tissue engineering and radiation decontamination. The first property is very important. This is the antimicrobial property of the antimicrobial activity. So we can check ki these types of waste product biomaterials are so antimicrobial property or not. If they are not so antimicrobial property, then this type of products are not used in as a scaffold or bone regeneration techniques. Uh, we can also check radiation wound model. We can this is the Wavatron 2 by exposure of gamma 2 radiation, and we can make a silver nanoparticle medicine. You can see the last lecture by Dr. Dipti Sharma ma'am. She also worked in this medicine, antimicrobial and radiation wound healing by AZNP. This is the you can check the seven group of the rats: control, F1, F2, F3, and F4 are the different doses of silver nanoparticle, that is 0.2%, 0.5%, 1%, and 2%. Other one is standard. We can use the already purchased from market and vehicle. This is no medicine will take this group, and we can check ki 0.2% and 2% in silver nanoparticle groups are the soda waste result, and this some are the histopathological results. We can see ki the epidermis do not rupture, and the epidermis is already maintained after the treatment of 2%, but we can see the control, and in vehicle the epidermis and dermis are rupture. We can also study the toxic effect of antimicrobial of carbon composites. Why we can check toxic effect of any material? Because when we can decontaminate the surface of the skin or the surface of the any material. This is very important कि these material are non-toxic, because if the material is toxic, then the double infection are caused, because first thing ki radiation cause also infection, and if the biomaterial who decontamination the radiation, these also affect the toxic. So we can secure the thing ki biomaterials are non-toxic, then it's used in decontamination efficacy. After we can check 14 and 28 days by OECD guideline, that is Organization for Economic Cooperation and Development guideline. This is two guideline by follow. One is accurate and second one is subaccurate. First, for accurate, we can check after 14 days, and for subaccurate, we can check after 28 days. And we can find that the carbon composites with a specific nanometer we can use. Then we can find no rats are mortality, normal pulse rate and breathing, normal food consumption, normal physiological behavior, normal body weight, normal urine, and no salivation we can found. After 14 and 20 days, 28 days, we can dissect the HD rat, Sprague Dolly rat, and we can check the histopathological results and biochemical results of the HD rats, and we can found ki one more thing, we can also check 14 and 28 days ki dermal irritation study and sensitization study, that is guideline followed by 106 and 110, and we can found as the drug does not show any irritation and any sensitization scores or any rate or any grades. This is the some biochemical parameters we can found after 14 and 28 days. The result shows the all group of the animal and all the carbon composites shows normal biochemical parameters, and it also shows that biomaterials is non-toxic. This is the some histopathological parameters from the HD rats of kidney, liver, spleen, and skins. We can found that to 500 mg per kg body weights rat and 200 mg per kg body weights rats, the skin of the skin, the layer of the skin, dermis and epidermis are found constant, but in the group of the control we can found the rupture of the dermis and epidermis layer, as well as we can check 2000 mg per kg body weight. We can just see that it's make a tumor-like structure in the histopathological results. And in the kidney cell, we can say the BC are the just common Bowman capsule cells are constant, but in control group, the Bowman capsule cells are also rupture. In also see in lungs, alveolar cell also constant in 200 mg per kg body weight and 500 mg body weight. But in 2000 mg per kg body weight and control group, we can see the alveolar cell are also rupture. When we can found that the biomaterials are non-toxic, then we can check the efficacy of the decontamination of radiation by the apparatus of France diffusion apparatus. In this apparatus, we can just stick the sample, stick the skin of the sample, and after 45 minutes, we can just apply the radiation operation, that is technetium, and 45 minutes, we can check by the atomic absorbance spectroscopy, and the result is very shocking, because the penetration of the biomaterial is just below 0.3. that is 0.3 ppm. That is very low penetration of the biomaterial, and we can show that key this is the very good biomaterial for decontamination. After that, we can check the decontamination efficacy by absorbing of the cotton. The cotton is dissolved in the carbon composite emulsion, and we can just wipe the surface of the skin on the rats, and we can found the control group that is only 10% absorbance. That is the cotton is dipped on water, and just wipe the radiation, and the cotton absorb only 10%, 10 to 15%. But when the cotton is dipped in the carbon composites, that is 1%, 2%, and 3%, and then we wipe the surface of the skin, then we can found that 75 to 80% radiation will absorbed by the cotton, and normal cotton just wipe the, then it is so 50 to 55% of radiation absorbed. After the result, we can conclude the material is non-toxic, and base decontamination efficacy, the penetration is less than 3.5 microns, and we can found no harmful effect of carbon composite, that is specific nanometer we can say already. And Dr. Mahesh Kumar Shah and Dr. Adesh Kumar Saini sir helped me in my PhD work, and we have already continued this work in my PhD. But this time we can derive the carbon composite by plant waste product, and we can also aim to use this product are used in sunburn or wound healing efficacy, like fracture bone wound healing models we can use. But my aim is future key, after searching the some website and NASA and site of the ISRO, we can found that key this type of biomaterials are or bone tissue engineering are used in very much in space stations. We can see these two type of space suits. First one is female astronauts, and second one is suits for male astronauts. And after putting this dress, we can find many types of medical problems, like women suffer hearing issue and advancing age, and do not display the waist two sides, hearing in the left ear. We can also see the clinical significance for visualization impairment. Female astronauts also susceptible osteostasis intolerance. We can found a problem in urinary tract infection for male and female also. Kidney stones are commonly found in females. In male, we can found the immune system are weak in the bones. But actually, this type of problems we can see, and also IM medical conditions, we can find 100 types of medical problems are seen in astronauts who live in 0G, means zero microgravity in space station. What is microgravity? In Earth, the microgravity we can say 9.8 meter per second, and this is so key 1G, and 1G means 9.8 meter per second, and at in the moon surface we can find 0.18 G, means only 0.72, sorry, 0.88 less than Earth's surface. And in the surface of Mars, that is Mars planet, 0.38 G. This is also very less. And International Space Station, the G is 0. And this is the problem of microgravity. We can found in the major problem. We can see some visualization effect. We can see the next slide due to microgravity. I hope slides is proper visible. My slide is proper visible. Hello. Hello. Okay. Thank you. Some visualization effect due to microgravity. When in the surface of Earth, the force look outside, and in the when we can see zero gravity, the force are repulsion each other, yani oxygen repulsion each other. And we can see the second figure. On the surface of Earth, the blood or the fluids inside the body flow continuously in upside and downside, that is in 1g, the normal fluid movement. No movement is changes in the surface of Earth. But when we look on the zero gravity, the we can see the visualization effect of the downside the figure. The fluid is only through move on side the brain. The leg is 0% fluid, and very minimum fluids are go inside the leg. And we can also see the last figure. This is the brain. The fluid towards the head cause increase internal pressures. And we can see that this figure also for cornea, fluid pressure cause optic nerve compression, flattening on the back of the eye wall, and optic disc swelling. This is the main effect of microgravity. Some other effects we can see in space, that is exposure to high level of radiation increase the risk of DNA mutation, degenerative disease, construct and life science lifetime risks of cancer. In case of bone, insufficient bone formation compared with bone restoration reduce the bone mass and bone strength, leading to an increase in fracture and osteoporosis risks. Due to immunological disorder, the immune system is weak in the ISS, International Space Station, or in zero gravity. Uh, Lack of gravitational loading and reduced muscular activity result in muscular atrophy accompanied by functional and structural alteration. We can see the main problem also caused by body fluids, that is headward shift of fluids. All fluids, maximum fluids are shifted towards the head, including blood, leads to compensation, cardiovascular system change, decreased leg volume. Leg volume is decreased due to the all fluids are shift towards the head. That's why the volume of the leg is decreased, and even long-term ocular damage causing vision problem. Uh, this is the space suit. Space suit is made by 9 to 10 layers. That is HT1 outer layer, and second layer is the micrometeoroid protection. Third layer is thermal layers. Fourth layer is micrometeoroid protection, registration net, pressure bladder, comfort layer, undergarments. And we can see that this suit is made by biomaterials. Then I hope the this type of biomedical issue are resolved by biomaterial making suits. This is the conclusion and future work: biomaterial derived radiation protection astronaut suits, microgravity and impulsion for wound healing, scaffold based tissue engineering in microgravity, space radiation decontamination soap and detergent kit. And this type of work I should work in future. This is my few publications. I am acknowledged our guides and supervisors. This work was conducted in DRDO INMAS, so I acknowledge our research team. This whole our research team in INMAS DRDO, and thank you. This is the session for question answering. If anyone want to ask any question,